it's when you're thinking temporal, across time, right? So multiple places, or multiple times, same place. So this would look like, this is a really ugly, so this is our dendrite, right? So we'll say that we have an axon synapsing here, here, and over here. So you're only getting activation at this synapse, right? You're giving excitatory once, excitatory again, and excitatory a third time, right? So same location, and everybody see this, same location across time. Happens once, it happens again, it happens a third time. Okay, so what's important here in terms of determining whether or not you're actually going to get summation is your time constant, right? So a longer time constant is going to essentially allow more easily for the postsynaptic potentials to summate. So you want a longer time constant because it's going to be more likely that you're going to get a summation and an action potential. Spatial summation is multiple locations. So all of these different axons right here are giving inputs, but it's happening at the same time, right? So instead of one location across time, you have multiple locations at the same time. So I would write that down and practice it so you guys remember which one's which, because I think those can be easy to mix up. And here, what's important in terms of deciding whether or not you're going to have summation is your length constant. So how far, essentially, can the axons be from each other, and you're still going to get summation of all of those different things. So the length constant would determine whether or not, for instance, this axon over here is included in the summation of these up here, right? So the more length you have, the more likely you're going to have other axons that are also adding in their inputs. So more inputs, obviously, is going to be more likely to summate. So again, our location is important. So the closer you are to the axon hillock, the more influence those axons are going to have in terms of deciding whether or not there's going to be an action potential. And so there are a few different types of axon connections that you can have. So this is our typical connection that we usually see. This is axodendritic synapses. So you have your axons synapsing onto the dendrites, and then those inputs are going to sum either temporally or spatially down into the axon hillock, which is then going to decide essentially if there's going to be action potential or not. You can also have axosomatic synapses, so this means that they're connecting onto the cell body, right, onto the soma. And so because these, let's say, these guys are connected over here, these three are going to have more influence than the two that would be up here because they're closer to the axon hillock. So let's say you have, for example, that these are excitatory and one of these is excitatory, but you have two inhibitory. Depending on a number of different factors, the inhibitory might win out even though there's less because you have two right here on the cell body, right? So that's going to depend on a number of different things from your, particularly things like your time constant or your length time constant, however these are summating. So location is everything, right? That's going to determine whether or not those inputs are going to contribute significantly to an action potential or not. And then finally, you can have axo-axonic synapse, which actually means that you're having one axon synapse onto another axon. And so this, of course, is not at all going to decide whether the axon hillock is going to trigger an action potential or not. But what it can impact is the amount of neurotransmitter release or if there's going to be neurotransmitter release. So when you think about that, if you have depolarization in this membrane right here, that could theoretically also depolarize this membrane. You could have the release of neurotransmitters or calcium that can then cross into this cell and impact the amount of neurotransmitter that's being released. So remember calcium when we talk about later chapters, calcium is going to be really important. Okay. All right, so we're gonna go pretty quickly through this because the main thing here you guys are gonna wanna focus on is gonna be your cyclic AMP pathway, right? And so again, we just talked about what a metabotropic receptor is. It's gonna be your indirect gating of ions. So what do you know? There's a list here, right? And so remember, Dr. Etheridge is super fond of lists because they make really good questions. So I would draw your attention to this and again, know well what the effects of a metabotropic receptor are going to be. We're going to keep going so we can get through everything. I think you guys are probably pretty good on that. I'm pulling out this stuff that's important here. So remember, you do not have to know this, which is really exciting. But you should know generally the cyclic AMP pathway. And so this is something you guys spent a lot of time talking about. So you should be comfortable with this pathway from top to bottom, right? So again, here's another list, right? So you should know exactly how the cyclic AMP pathway is initiated, how it functions as a second messenger, and the downstream effects, essentially. This one in particular, right? We don't need to know this whole mess of stuff, right? Cyclic AMP is involved in a number of different processes, but if you understand this list really well, you're going to be fine for cyclic AMP. Make sense? Okay. So this is a greater image of step 5. So does anybody have any questions about protein kinase A? Here's a lovely slide on protein kinase A if you need a refresher. So again, CAMP, CAMP is not the only second messenger, but it is the one that you guys need to focus on for the exam, right? So nitric oxide is another really good example. Carbon monoxide actually can act as a second messenger in many systems. Endocannabinoids are a big one, but these are not things that you guys need to know and memorize for this exam. This is a really excellent, this in red right here, is an excellent thing to remember. And so as an overall sort of overarching theme, it's important to understand why second messengers are helpful in biological systems. And so the main barrier to neurotransmitters particularly is going to be the cell membrane, right? And so if you have neurotransmitters that are lipophilic, so they don't want to come through that membrane, you have to have some other way to create downstream effects in the cell. So draw your attention, some mistake that we have like at least once every year is someone saying somewhere on an exam that neurotransmitters come into the cell and start doing things. They don't. Just like big letters, like your neurotransmitters are not going to come into your cells and start doing things, right? They're going to bind on the outside and either allow ions to come in or allow second messengers to work, right? And so the whole idea behind second messengers is sometimes you need greater activation within the cell, and a neurotransmitter can't do it because it can't pass through the membrane. Make sense? Okay. All right, again, you don't need to know the endocannabinoid system. These last few chapters quickly. Okay, so in general, know the importance of calcium to this system. You do not need to know the, so in general, importance of calcium, right? You do not need to know, we'll get there in a second, the disorders, right? So these are not necessary for you guys for this exam. In terms of calcium, know that they're needed to get transmitter release, right? So without calcium, nothing's going to happen. It's also important to understand exactly how neurotransmitters are released. So Quantal unit is a term that you're going to want to know, and so neurotransmitters are released in these units. These units are the same, you have the same number of neurotransmitter in each quantal unit for each type within a cell, you might say. And so the number of neurotransmitters within a quantal unit within a particular cell is not going to change, right? You're going to have the same number of neurotransmitter in each vesicle. And again, the more calcium you have, it's not going to make your quantal unit bigger, right? That's standardized. That's not going to change. What is going to change is the amount of quantal units of release that you're going to get, right? So more calcium you have coming in, the more vesicles you're going to have released from the reserve pool, binding to the membrane, and then fusing into fusing back out and exocytosing. It's not super important for you guys to understand the probability of release. However, it is important for you to understand where they're released, right? So the term active zone is going to be really important. I use the term postsynaptic density a lot when we were talking about the other chapters, right? So active zone is going to be your presynaptic membrane. That's where the neurotransmitter is being released from. And your postsynaptic density is going to be where your receptors are, right? They're going to be on your postsynaptic cell. Makes a lot of sense. Know how transmitters are released. So again, another term that's important for you guys to know is exocytosis, right? So the life cycle of a vesicle is going to be something that you should know. So vesicle is released by exocytosis and then reuptaken, you might say, by endocytosis. So it's recycled, that's a better word. So released by exocytosis and then recycled via endocytosis. So that's an important distinction. All right. So in terms of the structure of vesicle release, you don't need to know any of this down here, but you do need to know this stuff up here, particularly these guys within the snare complex, right? You don't need to know anything external about the ribs and beams. So for structure of vesicle release, you should know the role of synapsin. So this is what's binding the vesicles to actin in the cytoskeleton, and you require, this is the first place that you're seeing calcium interaction, right? So calcium has to come in for these vesicles to be released from the top of the reserve pool, right? All right, so on your vesicle, you have your V snares. So you should know the role of synaptobrevin. You should know that it's binding tightly to the T snare, and that's essentially what's anchoring the protein down into the membrane. Synaptotagmin has a really important role, right? So this is your second area where you're going to see calcium activation or calcium interaction. Know how many ions that it binds, and you should know the role in terms of it's activated, and then you have synaptotagmin swinging in, attaching to the snare complex, and then kicking complexin out of the way. This is a lovely image of all of these things. So on your actual cell membrane, you should know your T snares. And so you have syntaxin in your SNAP25. You do not need to be able to distinguish between them. You do need to know that they are T snares. You need to know, again, that they're going to be the things that are attaching to synaptobrevin and anchoring the vesicle to the membrane. Ribs and beams, you don't need to know. Do know that MUNC13 is one of your anchor proteins here, whereas your MUNC18 is the one that's actually going to be involved in the snare complex. So know the difference between 13 and 18. So MUNC18 is your sort of your safety pin for the snare complex, right? All right. MUNC13 is a scaffolding protein, so not important for you guys for this. So we talked about complexin. And then this is a really good image of all of those different things. So for the neurotransmitters, give me like a minute. For the neurotransmitters, you should know generally the different classes. You should know the different classes. You should know what it takes to be considered a neurotransmitter. And you should know the difference between small molecule transmitters and neuroactive peptides. And so be sure that you can distinguish between the two of those things. So the difference between small molecule transmitters and your neuroactive peptides. So those are going to be your hormones, right? So there are slides covering those. So there's a slide we talked about. There's a slide about the neuroactive peptides and it'll tell you the differences between those. So focus on that slide. Know that these are typically distinguished by their structure. So you have catecholamines with their catechol ring. Serotonin is going to be your indole ring. We didn't talk a whole lot about amino acids or ATP. And so in terms of the neurotransmitters themselves, know one really, really well, like top to bottom. I'm going to post the neurotransmitter sheets that I chose after class. And so look at that sheet, study it, know it really well, know it from top to bottom. You don't have to know all of the neurotransmitters that well. You should, however, don't just forget about the other ones. You should have a general understanding of their main functions. So acetylcholine is important for activation of muscle at the neuromuscular junction and for arousal and memory. Know then the function of serotonin, dopamine, et cetera, et cetera. Make sense? You should also know the drugs. Know how the drugs interact with the transporters. Know the difference between blocking a receptor and causing the receptor to not work correctly. And with that, I will let you go.